Bismillahirrahmanirrahim. In the name of God, the compassionate and merciful. My dear students of first year MBBS, the last and the fourth very important basic tissue is the nervous tissue. And in the first year MBBS, we are going to just have got a simple introduction about the nervous system. The more developed and the more important histology of the nervous system will be studied in second year MBBS, where we will study the histology of the cerebral cortex and the cerebellar cortex and the spinal cord. Here in this lecture, I am just going to tell you about the major things about the central nervous system. As I will start from the saying that the nervous system is actually, it is ectodermal in origin. It is developed from the ectoderm, and it is the neuron is the basic fundamental structural and functional unit of the nervous system, and a neuron is nothing but this is a modified epithelial cell, and it is the neuron, and there are neuroglia and some connective tissue which is present in the skull and the vertebral canal. As I have told you that this is ectodermal in origin, and it consists of modified epithelial cells, and these epithelial cells, they have got two very important and very well developed properties of the protoplasm. They are excitable, they have got the conductivity. In response to certain physical and chemical stimuli, the cells are excited, and they then gives rise to a response in the form of an electrical current which is passing over the cell membrane, and by the passage of this current, the nerve impulse is conducted over the cell membrane, and this wave of depolarization or action potential, it receives the information from the periphery, and it then orders the information and the actions from the central nervous system towards the periphery. So in this way you can say that the nervous system is composed of the central nervous system, there is a peripheral nervous system, and there is an autonomic nervous system. The central nervous system, again, it is composed of the brain and the spinal cord. And in the peripheral nervous system, we have got 31 pairs of the spinal nerves, and there are 12 pairs of the cranial nerves. You are knowing that the autonomic nervous system has got two divisions, the sympathetic and the parasympathetic division. This is the brain. You can see that brain is then again it has got certain parts, that is the forebrain and the hindbrain and the midbrain. Forebrain, hindbrain and the midbrain. I will tell you, I will start from the embryology of the nervous system. Let's talk about the embryology. In the third week of the intrauterine life, there appears a thickening at the mid-dorsal region of an embryo. This is an embryo, and you can see that at the dorsal side, in the mid-dorsal region of the embryo, there appears a thickening which is known as the neural plate, and this neural plate then it becomes a groove-like structure, and this is known as the neural groove. It is a slipper-shaped structure, and then later on this neural groove, its lateral edges, they combine together, gives rise to a tube-like structure which is known as the neural tube, and this neural tube is your future brain and the spinal cord. Initially the neural tube is open on both ends at the anterior neuropore and the posterior neuropore. Then in the 5th week of intrauterine life, there appears 3 primary brain vesicles. There appears 3 dilatations towards the cephalic end of the tube, towards the cranial end of the tube. There appears the prosencephalon, the mesencephalon and the rhombencephalon. The prosencephalon is the forebrain, the mesencephalon is the midbrain, and the rhombencephalon is actually the hindbrain. And the hindbrain is again, it is composed of the hindbrain is composed of the medulla oblongata, the pons and the cerebellum. Midbrain is a small piece of brain which is connecting the hindbrain with the forebrain, and forebrain is composed of the cerebrum in the form of the 2 cerebral hemispheres which are joined by a corpus callosum, which is a big commissure which join the 2 sides of the left and right cerebral hemispheres. And in the then again later on, the prosencephalon is further divided into the telencephalon and the diencephalon. Telencephalon becomes the cerebrum, and the diencephalon is your future thalamus and the hypothalamus. The mesencephalon remains as such in the form of the midbrain. The rhombencephalon gives rise to the 3 parts, that is the brainstem. Brainstem is nothing but this is the pons and the medulla oblongata. If we talk about the hindbrain is composed of this is the cerebellum and this is the pons and the medulla oblongata. These are the 3 parts of the hindbrain. Here I like to tell you that this is the neural tube which gives rise to all the parts of the brain and the spinal cord. The primary brain vesicles, they then continue downwards in the vertebral canal through the foramen magnum, and this part of the tube will be developing into the spinal cord. As it is a tube, so in the tube there is a lumen, and this lumen, it dilates towards the primary brain vesicles in the form of the cavities which are known as the ventricles. Rhombencephalon is known as the rhombencephalon because it has got a rhomboid shaped cavity which is the 4th ventricle. And then in the midbrain there is a small duct which is known as the cerebral aqueduct, and this cerebral aqueduct then enters into the cavity of the diencephalon, which is the 3rd ventricle, and 3rd ventricle is continuous with the cavities in the both cerebral hemispheres, which are the lateral ventricles. And in the spinal cord, the lumen of the neural tube will remain as such in the form of a canal which is known as the spinal canal. So स्पाइनल कॉर्ड के बीचों बीच एक कैनाल रन कर रही है, और ये जो न्यूरल ट्यूब का ल्यूमन है, ये जिस तरफ डायलेटेशंस अपीयर होती हैं उस तरफ ये कैविटीज़ बनाता है जिनको हम 4th वेंट्रिकल, 3rd वेंट्रिकल और लैटरल वेंट्रिकल कहते हैं. एंड दीज कैविटीज एंड कैनाल्स इन द ब्रेन, they are lined by a simple columnar epithelium which is known as the ependyma, and these are the cavities which contains a fluid which is known as the cerebrospinal fluid. So cerebrospinal fluid is a very important fluid. We need cerebrospinal fluid sometimes to diagnose the diseases of the central nervous system. So we do a lumbar puncture and we remove the cerebrospinal fluid, and we are not going to pierce the spinal cord, but here I will want to tell you that the brain and the spinal cord is the gift of the God, and God has wrapped the gift in 3 meninges. There are 3 membranes which are covering the brain and the spinal cord, and these 3 dense irregular connective tissue membranes, they are known as the meninges, the dura. So nature has provided the brain and the spinal cord, there are three membranes which are made up of dense irregular connective tissue, and these membranes are the dura mater and the arachnoid matter and the pia mater. The dura mater is the membrane which is outer, and middle one is the arachnoid matter, and one which immediately surrounds the brain and the spinal cord is called the pia mater. And as you can very well imagine that there will be a space in between the pia mater and the arachnoid mater that is known as the subarachnoid space. And in between the arachnoid and dura we have got the subdural space. Subarachnoid space is the space where circulates the cerebrospinal fluid. So we will take the cerebrospinal fluid by lumbar puncture, by piercing the in fourth and fifth, in between the fourth and the fifth lumbar vertebrae. We will put a wide bore needle and we will remove from the subarachnoid space the cerebrospinal fluid, and we will take the cerebrospinal fluid for the pathological examinations, for the lab testing. And as you know that the spinal cord and the brain, it is composed of the neurons and the neuroglia. As regards the composition of the brain and the spinal cord, we have got the actual functional portion, that is parenchyma of the brain, consists of millions of neurons. Neuron, which is a basic fundamental structural and functional unit of the nervous system. This is a modified epithelial cell, and this cell has got the property of excitability and irritability and conductivity. But there are also cells. If there are millions of neurons, there are billions of neuroglia, neuron binding cells, neuron glue. न्यूरॉन को जोड़ने वाले सेल्स को न्यूरोग्लाया कहते हैं. न्यूरोग्लाया आर द स्पेशलाइज्ड कनेक्टिव टिश्यू सेल्स प्रेजेंट इन द नर्वस सिस्टम, एंड दीज आर नोन एज द एस्ट्रोग्लाया, द ओलिगोडेंड्रोग्लाया, माइक्रोग्लाया and the ependymal cells. These are actually the cells which provide the nourishment and support to the nervous system, that is the neurons. And here I will introduce you about the grey matter and the white matter. In the brain and the spinal cord, where there is the collection of the neurons, that area is seen with the naked eye as a greyish area, and we call it the grey matter. And where there is the collection of the fibers, these fibers, because the most of the fibers are myelinated, and the myelin sheath, I will tell you later on about the myelin sheath. So these myelinated nerve fibers, they are seen white in color. So the collection of the white matter is actually nothing but this is the collection of the fibers, and the grey matter is the collection of the nerve cell bodies. And here I will want to tell you that the distribution of the grey matter is on the outer side in the form of the cerebral and cerebellar cortex in the central nervous system in the brain. And in the spinal cord, the grey matter is on the inner side and white matter is on the outer side. The grey matter in the spinal cord is in the form of a central grey column which is lying in the center of the spinal cord in the form of a column-like H-shaped structure. And around this H-shaped structure, that is the central grey column, we have got the white matter. So the grey matter and the white matter, this is another very important concept. In the nervous tissue, there are two types of cells: the nerve cells, which are the neurons, they receive and transmit the impulses, and then neuroglial cells, more numerous than the neurons, they support the neurons in various ways. There are capillaries present, there are no lymphatics. Another very important type of the tissue is actually the connective tissue. As I have told you that the spinal cord and the brain is covered by the meninges, which are the dense irregular connective tissue membranes. Then let's talk about the neurons. Neuron is a very specialized cell, and you can see this is one of the neurons. It consists of three components. There is a nerve cell body, there are dendrites. Dendron mean tree, and these are actually the cytoplasmic extensions around the nerve cell body in the form of a tree. दरख्त की शाखों की तरह फैले हुए प्रोसेसेस को डेंड्राइट्स कहते हैं. And then there is a very long cylindrical process which runs downwards, and this process is of uniform diameter, and it ends up at a swollen end which is called the terminal. So there are three components of the neuron: the soma, the dendrites, and the axon. And this type of a cell is a very important cell, and with all the usual organelles which are present in the cell. Let's talk about the nerve cell, that is the neuron. The neurons are of different types. We can classify the neurons here. There are multipolar. There is a morphological classification, there is a functional classification, there is the neurons according to the length of the neuron. Another classification is the functional classification. There are sensory neurons, there are motor neurons, that is afferent and efferent. As regards the structure of the neuron consists of a nerve cell body which is known as the perikaryon, and perikaryon is very important. The area around the nucleus is called the perikaryon. In the nerve cell body there is a neuron, in the nerve cell body there is a nucleus which is euchromatic. It is pale staining with a lot of dispersed chromatin material, but in the center there is a very prominent nucleolus, and it gives rise to an owl's eye appearance. The area around the nucleus is called the perikaryon. Perikaryon or soma. This is because of the fact that in the perikaryon there are all the usual organelles present, but very important thing to mention here is the Nissl granule or the Nissl body, and there are neurofibrils in the form of the microtubules and the neurofilaments and the microfilaments. The Nissl bodies are nothing but these are the heaps of or the clusters of rough endoplasmic reticulum which are studded with the ribosomes and the polysomes, and the Nissl bodies are actually they are lying at the proximal ends of the dendrites. The area from where the axon emerges, that is known as the axon hillock, and axon hillock, this is the axon hillock, and from the axon hillock we will see that there are no Nissl granules present. Nissl granules, these are the Nissl bodies or the Nissl corpuscles which are lying at the proximal ends of the dendrites, and this area, this pyramidal shaped, it is hill-like area called the axon hillock, from where the axon emerges is devoid of these. So this is the nucleus which is present in the center of the cell with a very prominent nucleolus. So this is called the owl's eye appearance. Then in the cytoplasm there are all the usual organelles. There will be the Golgi body, there will be a lot of ribosomes, there will be a lot of lysosomes, the like lipid and granules and the other pigment granules, but the most important thing is the presence of the Nissl bodies. Melanin in certain regions, for example in the substantia nigra, and it may be byproduct of the synthesis of dopamine and noradrenaline. There is lipofuscin more in older adults, crowded organelles and nucleus to one side and may affect the neuron function. There are lipid droplets, may be energy reserve or the result of the metabolism. The cytoskeleton of the perikaryon, it is made up of the microtubules which are 24 nanometers. There are neurofilaments which are 10 nanometers in diameter. there are intermediate filaments. Neurofibrils possibly are clumped bundles of the neurofilaments. So this is the perikaryon. You can see the owl's eye appearance, nucleus, all the usual smooth endoplasmic reticulum, the ribosomes, the lysosomes, the Nissl substances, and you can see the dendrites, which are these cytoplasmic extensions around the body. And there is a long cylindrical process which is of uniform diameter and runs downwards. This may attain a length of 100 centimeter, and this is the axon. Neurons are a variable size. They are 5 to 150 microns in diameter. Their shape is spherical and sometimes angular. An axon, which is myelinated or unmyelinated with terminals and synaptic junctions, it is up to 100 centimeters in length. That's the reason I have said that the neuron is the longest cell in the body. Let's talk about the dendrites. These are highly branched, just like the branches of a tree. Receptive surface for the synaptic junctions. Tens of thousands of synapses on large dendrites. Dendritic spines are located on the surface of some dendrites. Spines diminish with age and poor nutrition. Then axons. Axon projects from cell body at axon hillock. Some axons are up to 100 centimeters in large animals. Axon hillock, a pyramidal shaped region of the soma that is devoid of rough endoplasmic reticulum, that is, there are no Nissl bodies present there. Initial segment is a portion of axon from its origin to the beginning of the myelin sheath, referred as the spike trigger zone. At spike trigger zone, summation of excitatory and inhibitory impulses occur. Then there are collateral branches. The axons may be myelinated or unmyelinated. Here I will like to tell you these are the myelinated segments. This is the node of Ranvier. The area between the two nodes is called the internode. This is the initial segment starting from the axon hillock up till the first segment, which is myelinated. This bare area is called the initial segment. Then these are the myelinated segments, which are known as the nodes, and nodes of Ranvier and are actually the interruptions. It is just like ये बिजली की तार की तरह होता है. बिजली की तार में जो कॉपर वायर होती है, जो लाइव होती है, उसके ऊपर एक इंसुलेशन चढ़ी हुई होती है और वो प्लास्टिक इंसुलेशन होती है. आप इमेजिन करें अगर हम उसको जगह जगह से काट दें, निबल कर दें और उस जगह से उस कॉपर वायर को नंगा कर दें, तो उनको हम नोड्स ऑफ रैनवियर कहेंगे. सो दीज आर द नोड्स ऑफ रैनवियर. दिस इज अ नोड ऑफ रैनवियर, दिस इज अ नोड ऑफ रैनवियर, एंड एरिया बिटवीन द टू nodes is called the internode. The longer is the neuron, the longer will be the internode, and thick will be the myelin sheath. Let's talk about the classification of the neurons. There is a morphological classification, that is, there are multipolar neurons and the bipolar neurons and the unipolar neurons. Multipolar neurons are the most common type of neurons. From the multiple poles of the nerve cell bodies arises the many dendrites and there is a single axon. The bipolar neurons, these neurons are located in the vestibular and cochlear ganglia. They are actually the spindle shaped cells. They have got two poles. On one pole there is an axon and on the other pole there is a dendrite. Then there are unipolar, also known as the pseudo-unipolar. These are located in the dorsal root ganglia and the other ganglia of the sensory nerves in the The functional category of the neurons, there are motor efferent neurons conduct the impulses to the muscle and the neurons and the gland. Then sensory or afferent neurons which receive the information from the periphery, and there are interneurons which form the local circuits. So this is the picture which can show you. This is a multipolar neuron, this is a pseudo unipolar. इसलिए कहते हैं इसको, देखो, this is the nerve cell body. It gives rise to an axon which immediately divides into two in a T-like fashion, bifurcates and gives rise to an axon and a dendrite. So this is a spindle shaped cell, a globular cell which gives rise to the dendrites on